He's on there now. All right, guys, we're going to get started. So welcome to the second meeting of neuroscience and deep learning. And so here's just some different links. I'm sure you guys have already checked them out, but you have we're keeping a blog at that website just to put up some information about what we're doing and stuff. And then we also have a GitHub repo where we put up the slides and papers and stuff that we're talking about. And if you want to tweet us, here are those handles and stuff. Now we have Andrew working with us. He's working in the shadows behind the... We're not sure what Andrew's going to do yet, but I'm sure it's going to be useful. So it'll be nice to have his expertise helping us navigate. So just to kind of put things in perspective with our calendar, so around a month ago we had our first meeting. Arna and I introduced some concepts about deep learning, and so today we're here, meeting 2, March 20th, and we're gonna have a presentation where we're gonna talk about some potential deep learning application, and so we're not sure exactly what's gonna come of it, but just to give you an idea of how deep learning can be used. And then we're going to go into our paper that we sent out, and I'm sure everybody read, and there's a lot of questions. So we didn't get a lot of people to sign up to present. So so this is just the agenda, like the calendar for the upcoming meetings. So probably at the next meeting, we're going to have somebody give a presentation of his or her work. We haven't decided yet on who that person is going to be, but our idea was to get everyone to suggest a paper that they would like to discuss. So between meeting 2 and 3, so between now and April, if you can email us a paper that you want to discuss, and it can be on deep learning and neuroscience. It's very broad what we're doing. So if you think something is interesting to you, then you can send us. And then between meeting 3 and 4, we would send out the list of papers. Like, I don't know how many papers, 10 papers, and then we can all vote on a paper and then sign up for the 4th meeting in May. So yeah, we're going to be sending out emails about all that stuff. So, and then as you can see, like all this stuff is still to be determined. So if there's something that you guys want to do, just let us know. If you want to do more journal clubs, or if maybe somebody you know wants to present their work, send us an email. All right. So with that, we're going to get right into the presentation. So who's going to be going first? Okay. Okay, so we have today the privilege of hearing from Dr. Mathilde Chenot. She is a research assistant cell biologist with broad experience in molecular and cell biology research. She has a PhD in cell biology from the Université Pierre et Marie Curie. She did a 5-year postdoc training here at McGill studying vesicular trafficking. And after her, we're going to see some cool videos and stuff on the data set presented by Relena Thomas. She received her PhD here at McGill in neuroscience. She did a 1-year postdoc with Dr. Maurice Chacron. And now she's starting her 2nd year postdoc with Dr. Edward Fawn at the MNI, applying computational analysis techniques to biological questions. Primary research project is to analyze single-cell RNA sequencing data of human cell-derived mini-brains. So yeah, let's hear from Mathilde Chenot. Is it okay?
Yeah, okay. So hi everyone. So it's going to be like a presentation together. Actually, it's not going to be 2 different presentations. And the thing is that I'm really a cell biologist. I know pretty much nothing about computer and even less about deep learning and AI. But I think it's a cool way to learn how to communicate together and maybe make more things together. So Ralina asked me to introduce you the cell biology and a little bit of the project before going into data and what we can do together. So I'm working on ALS, so maybe you know, maybe you don't, but like, what is ALS? It's also called Lou Gehrig's disease. In Europe, it's mainly called motor neuron disease or maladie de Charcot in France. It's a terrible disease. It's a neurodegenerative disease that is progressive and it's terminal. There's no cure right now for that. In Canada, 2 to 3 people are diagnosed every day, and the life expectancy is 2 to 5 years when you receive your diagnosis. But we see more and more lately some people who can live longer with that disease, like you all heard about Stephen Hawking, for example, that lived like almost 40 years with the disease. Mainly ALS is sporadic for 90%. You only have 5 to 10% who are familial cases. And the disease is a big burden because it costs a lot of money for people and family where there is someone with ALS, because you need to buy very expensive equipment. People will end up in a wheelchair, electronic wheelchair. They will need to have a system to communicate by eye movement. So we really need to find cure for ALS. So now, like, just a little bit of what happens. So in healthy individuals, you will have like upper motor neurons localized in your brain that will communicate with your lower motor neurons localized in your spinal cord. And those lower motor neurons are making connection with the muscle. So if you want to make a movement, your brain gonna send the information to your lower motor neurons that will connect the muscle and give them the order to contract so that you can do a movement or grab something or like run away, those kind of stuff. If you have ALS, there is a progressive degeneration of your upper and lower motor neurons, and so the contact with the muscle is not made properly, and eventually the muscle is gonna degenerate. And so you will lose the ability to do simple tasks that you do in your life, grab object, talk, walk, and eventually people with ALS died from respiratory failure because the diaphragm muscle is not working properly and they will not be able to breathe anymore. So it's a terrible disease, like I said, and unfortunately there is only in Canada 2 drugs that are available right now, one that is available for 20 years. The other one just got recently approved by the Canadian Health Agency, but that's it. And it's a disease that is known for a lot of years now, and there's lots of research made, so we don't really understand why there's no cure yet. It's a big challenge. And one of the reasons we think is that we use animal models. So don't get me wrong, animal models are absolutely necessary, but there has been a very promising result with rat, mice, C. elegans, zebrafish for ALS, but as soon as you transfer those molecules into human, the clinical trial failed. And it costs a lot of money for Canada, for government, for clinical trial, and it's a lot of like false hope for patients. So one way to try to go over that is we've been lucky that almost 10 years ago, like a researcher called Yamanaka identified some factors where by using like easily accessible human cells, you can de-differentiate those cells into what we call stem cells. So they're not embryonic stem cells, but induced pluripotent stem cells. And from those iPS, by giving them different factors, you can pretty much give birth to any cell that is in your body. So that was the only way now, it's the only way that we can access human cells to study the disease in a dish. Because it's not like cancer where you have a tumor, it's easy to get the tumor removed, put a little bit of cells in a dish and study the mechanism. If you have brain disease, we're not gonna go get like a chunk of your brain to analyze what's going on. So this is a really big step for us. And so when you have those iPSC from control and from patients, you can actually study them. You can correct the different mutation by doing CRISPR-Cas9, and then you can differentiate. In our case for ALS, what we're interested in is doing spinal motor neurons because those are the neurons who degenerate. We are always also making muscle to recreate the neuromuscular junction and glia, that is the support cells for the neurons. Then when we have that, the idea would be to study the disease in a dish. We can do toxicity testing for preclinical trial and drug screening. And this is just an example of the cells that we're using. So those are bright field pictures. This is a bright field picture of iPS. And so what we're doing, basically, we characterize the cells by doing immunofluorescence or PCR to see what gene they express at what time. And to make motor neurons from iPS, it's a pretty easy protocol where basically you feed the cells at like specific time point with different factors that mimics the development in your body when you're growing up, like when you're an embryo and so we have like a step in between that is called NPC for neural progenitor cells. They look like that, and same thing, we characterize them with immunofluorescence. And from those NPC, by giving them growth factors, we are able to differentiate them in motor neurons. And the idea would be to compare the one from control and the one from patient to see what's going on, what the differences are, and so on and so forth. And eventually do some drug screening on them. This is a picture of what the motor neurons look like in the dish after 4 weeks. And they express like the specific motor neuron marker HB9 or ILET1, exactly like the one you have in your body. So it's a way to check that everything is fine. And they form after like 4 weeks here, but even a little bit before, they form those clusters. And that is pretty hard for us then to analyze with the tool we have right now. So this is a movie and that's what we're gonna talk about, I think. It's a movie of NPC that I plated, and I just image them with bright field pictures. We have like a system in the incubator that can take pictures over time, and then we recreate a movie out of them. So you will see motor neurons growing live, basically. So it's a little bit long. It has been imaged over like, I think 3 weeks, this one. So that's what they do. They extend their axons, they communicate with each other, they retract. All the cells' body come together in clusters. And so the idea, but like Ralina is going to take over, is like, what can we do with that? Can we use deep learning to analyze, to see if we can see difference between control and patient cells, and those kind of things.
So I showed this video or one like this, probably the same one, to Andrew and Ricardo, some other guys before, and saying like, whoops, how do we go to the, oh. So there's actually, the data we have, this video is composed from imaging sessions over 6 different sessions of imaging, and then it ends up being 1,000 frames. And there is actually 48 different fields of view, so we could create 48 of that same video from the same cells. But we don't have the equivalent video from a control cell, or from a patient cell line, to just use a deep learning to compare like unbiased without any feature selection. But we thought it was also interesting to see if could we create more of like a reiterative training AI that could recognize cells, track them over time, and then we could calculate things from there, like how many cells go into the cluster, how many cells die, what are the kinetics of movements of the cell. All of these things would be interesting quantifications for us, but we don't have any way to do that, so there hasn't been any motivation to create more of these videos. We can easily create these time series. Well, easily, it's not that hard to seed the cells, but it is actually a lot of work and a lot of money and a lot of time, so there's no motivation to do it unless we have a good analysis at the end. So we kind of go back and forth between those two places of, well, it would be better for analysis if we have more data, but we don't want to create more data without a way to analyze it. So what we have right now, like I say, is these 48 videos that are all the same times, over the same number of frames, but the time frame between them for the first 4 imaging sessions is 20 minutes, and then later it's 40 minutes. So if you're tracking a cell, it's going to have maybe more movement, but it also goes over the differentiation of the neurons. So at the end, they're not moving nearly as much as at the beginning. And then there's a lot of other videos. So there's another set of videos of neurons of differentiation, just from a different experiment where they're imaged every 30 minutes, and there's 144 frames, or sorry, fields of view for those ones. So yeah, I just open for discussion. Any ideas or thoughts on that? Questions? Yeah.
So on the previous slide, you had different characterization
Yeah.
that you wanted to do. If not for deep learning, what is the best solution right now?
Well, what are we doing right now?
If not for deep learning, what is the best way to do this right now?
Like manually, but we don't do that. The only quantification that we have done from, not even from these videos, but from the last time point where they're already in the clusters. So we have cells that are stained with immunofluorescence markers, and then we can, Eddie actually has made an ImageJ macro that traces the clusters, so you can count the size of the clusters, measure the distance between the clusters, and then you can locate the cells that are not in clusters and see how many of those have your motor neuron markers. So we have no information or any way to analyze the kinetics of the cell movement. We can manually go through the video and choose the frame where we say, okay, this is now a neuron, but we are not going to do that. I mean, that's something that we could do, but that's not practical to do, and it doesn't gain us enough valuable information to say oh, this cell line most of the time is a neuron at I don't know, the 15th day, and this one's a neuron at the 16th day. That's not that useful of information to put in hours and hours of work to find out. It could become useful combined with other information. So right now, our main, the big picture is to have abilities to characterize cell lines, to find out how much variability there are in control human cells from one human to another human, and then compare that to how different they are from the cells from people with ALS. And then the long-term goal is to be able to have something scalable for a drug screen, where you can say which drugs recover the disease phenotype we don't know much about these, so the aim, starting point has been to characterize them.
Yeah,
So that's
yeah. Because even without talking about identifying difference between patient cells and control cells, right now we don't have, we're working on it. As I said, like we can correct the mutation so that you have exactly the cells from the patient that you correct. So you have the same genetic background to compare. And that's the best way to do it. But it's a very long process to do. So right now what we're doing is that for the control, we have like basically healthy volunteers that will give their blood or their like skin cells that will reprogram. So we are comparing patient and control, but what is the best way, how you would select the best control for your like disease line? Is it gender? Is it age? Like all of that, we're working on that, but maybe like it's just ideas like that, but maybe by characterizing them deeply, we can actually take a disease line, put it in the computer and say, okay, select me the best control that would match that line, you know, for example. Those are things that we're just thinking about and yeah.
I guess this is a very open-ended question. You know, it's very exploratory at this point.
Yeah, it's
And
very exploratory.
There's nothing solved. It's just there's some cool data set.
Yeah,
Deep learning could help.
early days of well, we did this, and now we're thinking about it and what could we do.
I mean, we're still doing stuff like basic cell biology and stuff with that. So it's we're working on it, and but it's just like, okay, can we? Because it's all about now deep learning, AI, and how like Montreal is a big hub for that, and we can like. I think it would be cool that if we can interact a little bit more and bring expertise, and I think it's all about that. So I don't know if you have ideas or.
Yeah.
So it's just because we have to take out the cells to change media and put it back. So we have a system that can record position, but like you know, the plate is an incubator on the stage that is like mobile and stuff. So you take out the cells, change the media under the hood because it has to be sterile, and then put the plate back. And so the thing, it's actually so, and there's nothing we can do about that. So
Even when you don't change, sometimes the autofocus changes on its own. Yeah. I think so, but then you'll get out of focus picture.
because then you let it for like days without touching anything, and like it's an incubator with CO2, and the temperature is like, and it's an electronic stuff inside, so there's always like a little bit of variation anyway. So restart the.
Yeah.
that's good because that's a way. If we want eventually to use deep learning, it's good to have your input on what's wrong with our like image or stuff, so that we can correct before like acquiring tons of pictures and so yeah.
Okay. Yeah, so
Thanks for your input, guys.
thanks for coming on the presentation.
Yeah.
Arna, do you want to come? What about Raymond?
Yeah. Do you want
Raymond
to?
will introduce it.
All right. Yeah. Sure. So we'll be starting with the paper discussion. So we have Raymond who signed up for presenting the introduction to us and the setting of the paper. So thank you, Raymond, for signing up.
Hi.
Do you have your own slides that you want to connect to?
Can I?
Yeah. Sure. You
How
can.
does it work? You have this.
You have. Which adapter is this?
HDMI?
Don't you have to put it in here? Oh, it works. Okay. So let's see. And yeah, just a second. Yeah, probably. You have to change screens or something.
Yeah, I don't know.
Probably. Oh.
Do that. I don't know how to do that.
Is Tony there? No. Yeah.
If not,
Can you
I try to see if I
Copy it
can.
here? what? Is it
I'll
coming?
email you. Maybe.
Do you have it on Google Slides?
Yeah.
You can log in.
Oh, you can log in.
Just do incognito,
I sign out.
or you can just do incognito and sign into yours.
What is that? Sorry. Ah.
Okay. So there you go. Yeah.
Okay. Good question. What is the password? This is just too many accounts to manage. Sorry for this. I don't know my.
Paper.
Hmm?
Paper for the next meeting. For the
About the sending. Yeah, you can say something too.
Aha. Oh. What does it need? Do you have your phone? Yes. Sorry.
Tap 9 on your phone.
This device not recognize.
He's not there, huh?
No. Tap 9 on your phone.
Tab 9? Oh.
Tap 9.
Open the Google app.
Tap 9.
No, it says
Oh,
open the Google app. I don't even know if I have a Google app.
You don't?
What is the Google app?
Oh wait. It should send you a notification. Are you connected to the Wi-Fi?
Yes, yes.
Oh.
Don't
Can you save?
send a notification.
It should
Open the
send a
Google app.
Can you
notification.
save this?
Yeah, maybe I will just
Yeah,
export
maybe
the PDF and.
Yeah.
Sorry.
Yep. Yes. Oh, we have the room till 2:30, but yeah. Okay. Yeah, we might run over a little bit, yeah.
Okay, I have sent it
Did
to
you
your
have slides?
email.
I added it.
What?
I added them on your.
I sent it to your email.
Great.
To whose email?
His. Well,
Mine?
yeah.
Or Nick Ricardo's.
Just your email. Anon. Yeah.
Or
Okay.
wait, you're already signed in, right, Ricardo?
Not my Gmail. I mean, I'm Gmail.
Yeah, I'll forward it to your Gmail.
I think it's.
What is your Gmail again?
RicardoPI@Gmail.
Ricardo PI at Gmail. This one?
Yeah.
Yeah, I sent it to you. this, right? RSCRDO Gmail?
Yeah.
Oh. I don't know why that's. Did you get it? No?
No.
Doesn't show on my outbox.
Okay, what is your email? I will send it directly to you.
RicardoPI@gmail.
You
Yeah,
just
that one
put
is fine. That one is fine.
The one is fine?
But you have to log into your Outlook again.
Okay, it's okay.
I think it's sent. Yep. Is it attached? Okay.
It seems Gmail doesn't work as well. Okay, so we.
So how? I just have to control from it.
Yeah.
Uh,
Maybe you can present from there itself. Yeah, better. Okay.
Presentation.
Okay, that's fine, right?
Okay.
You want this, right?
Yeah.
I'll let
Okay.
you introduce yourself.
Okay. Okay, hi everyone. Sorry for the delay. My name is Raymond. I'm actually a PhD student at McGill at Mila in computer science. So my background is not neuroscience. So please forgive me if I say the names wrongly. So basically, I agreed to do the introduction because most of the things were actually about deep learning and CNNs. So before I came to Montreal and McGill, I did my master's in Germany in computer science and also working in computer vision. So the introduction part was probably the easiest part for me. So I will just quickly say that I will just cover the CNN and feature extraction, and then the task, which is about object recognition in the paper, and then experiment overview. So I think everyone knows what is CNN here. If not, I will just briefly summarize. In CNN, you have the first few layers of convolution, and then these are just sliding windows, and then you extract. This is where you try to extract features, and the pooling is just to do downsamples so that you make your feature space smaller, and then the fully connected layers are just for you to do your prediction, and then finally you just classify them based on the different classes. So here we have some animals, and what is interesting for this paper is that for the features abstractions, so in deep learning, you don't have to explicitly handle the feature extraction, meaning that you don't have to do features engineering. So these features are learned end-to-end via a loss function. And what is interesting is that these features are in a way similar to these Gabor filters. They are probably observed in neuroscience. Correct me if I'm wrong. And these are usually observed in the lowest level, so layer 1, which is closest to the input images. And as you go more into more deeper, where you try to get closer to the classification area where you have the fully connected layers, where you have all these features that are very closely related to the classes of objects they're going to classify. So yeah, in the paper it says that similar to what is known about response properties or the neural populations in V1. This is, I guess, the cortex stuff. Okay. Now, the task that they try to address is object recognition. So, so like I said, so right now the state of the art is to use deep learning because you don't have to handcraft the features and you can, and the performance is really good. And the challenge of object recognition is that the objects you have to train an algorithm that's invariant to scale, translation, and deformation. And these are usually not easy because of sometimes occlusions and things like that. So a standard computer vision task is to, for the algorithm or the AI to detect what object is there and to draw a bounding box around the object. How good the algorithm is depends on the area of intersection of the bounding boxes. So in the paper, what we have is that they want to combine CNNs with electrophysiological measurements of neuronal activity, such as EEG and MEG, to probe the dynamics of the object processing in the human brain. And then they have so this was based on some previous work. And right now, their work extends on this and uses an encoding model framework to probe how CNN-based representations are expressed in space and time across the cortical surfaces using MEG. Now, in the experiment, there are 15 participants, and each of them are presented 1,000 images of natural objects. The MEG data are recorded. A random subset of 50 images are repeated 10 times as validation sets, and then the majority of the 950 images were kept as for like test data estimation sets. So basically, the participants are not asked to do any specific tasks, but just to passively view the images. So a quick workflow of the diagram as follows. At the start of trial, they are shown a pre-stimulus fixation for 400 milliseconds, and then they are shown the object, which is the stimulus, and then again, they are shown a post-stimulus fixation, and then there's a blink phase of 1,000 milliseconds, and then that's the end of the first trial, and the trial continues. Right. And then the computational goal is to arrive at the invariant cortical representations of the objects, which is believed to be realized by the transformation of the retina input through a series of nonlinear processing steps in the ventral stream. The resulting representations are assumed to reflect in the characteristic patterns of the brain activity, which is discovered using the candidate representation to predict the brain activity based on this encoding model framework. Now, to link the neural networks to the patterns of the brain activity, I have to say this. The authors, they use a very simple deep learning architecture, which is very close to similar to the one I presented in my first slides. So they consider these neural networks and the features that's learned, and then they model the cortical source activity from the MEG sensor using the source reconstruction on the individual brain surface models. And then for each participant, they develop a source-wise encoding model to predict the model source activity using these CNN's representations. The encoding model will then uncover when and where the cortical activity is similar, and because of this encoding process, we can also use it for this decoding, where the most likely stimulus is selected from some preset, some set of candidates based on the observed brain activity. Okay, I think that's all from my part. So, yeah. No.
We're gonna go into the methods now.
So I'm just.
So maybe we can have the discussion afterwards. But if you want to say something.
No. So I think it's best that you hear the rest of the because they're going to show how the whole thing works.
Okay. So this is the paper again. Yeah, actually, Raymond went over a few things that I was going to present. So, it's a lot, Raymond. No, it's good. I have to talk about less. So, yeah, just to reiterate, there's a lot of images that were presented to both the participants and the trained convolutional neural network. And they were presented in this way. As Raymond said, there was the estimation set, the validation set, and they were presented in 10 blocks of 145 images. And between each block, the people making the recordings, they talked to the participants to make sure they were okay. So a couple of things that were unusual when I was reading the paper is that they say that they rejected some trials based on pre-processing, and then they average up to 10 trials per stimulus. But they don't really say if there was a minimum number of trials per stimulus. So I thought that was a bit strange, or maybe they talk about it later, but and then they asked the participants to avoid scheduling MRI recordings during the previous few days. So I'm not very familiar with MEG, but I don't know how MRI recordings can affect MEG recordings up to a few days. So, yeah, go ahead. Oh, okay. And it needs like you need a few days for this to resolve, or it's just standard? Okay. Okay. Cool. And then I just like the term non-magnetic clothes, but so I guess just like jeans, like with metal components or something. And then participants were informed of the passive nature of the experiment. To me, it seems like a little subjective as to when the person is performing the passive task, if they're thinking about something or not. It seemed to me a little bit hard to monitor exactly what they're doing or thinking about as they're looking at these images. And then they were asked to, between the blocks, they were asked to adjust their current position, but they don't really say how they need to move, how this affects the MEG recordings. But so, anyways, so they use this MEG, the CTF 275 channel. So this is what the MEG machine might look like, and then you have all these sensors around the head recording magnetic activity. And then they took after the MEG, they did a structure MRI for analysis, and from each MRI they constructed this fancy mesh around the cortex, and this was used for the source localization. So they used the mesh, the vertex on each of the triangle. So like here. Each of those points were used for source localization, for source reconstruction. So it's a way to map the MEG signal onto the brain. And they started with this many number of vertices per hemisphere. Then they downsampled, and then they removed the midline points, and this is what they ended up with to cover the brain. So this is sort of a schematic for what they did. They started with the number of sensors, 275. And then on the right you have the number of dipoles that they use, which is 7,000 something. And here this is how you map. This is how you go from one data set to the other. So you have the MEG signal on the left, and then to get to the activity of what's going on in the brain, you have this ill-posed inverse problem, because it's hard to make this transformation. But to go the other way, it's the forward model where you can, if you know the activity inside the brain, you can then estimate what the activity might look like at the MEG. But it's not. The reason why it's ill-posed is because there isn't a one-to-one correspondence. So multiple brain states can generate a similar signal in the MEG or EEG. So for each image that the participant viewed, then you get a signal at each of these sources or dipoles defined by the mesh here. That's described like this with a Y subscript ST. So these are source responses. So for each image N, you get a source response YST. And then what Andrew was asking about was they took a pre-trained convolutional neural network. So the parameters were already estimated. and they just pass the image through the neural net. So you have the image, and then you can just, without changing the parameters, you just pass the image through, and then at each layer you get a specific value, specific feature. So for each layer of the convolutional neural network, you get another signal, which is a feature representation of the layer. So here you have, this architecture has 8 layers, L equals 8, and each feature per layer is of size m by 1, depending on the layer. So because of the architecture is different, then each layer will have a different size. Will be different for each layer. And then this is all the fancy math that they use to correlate those features, the feature representation that they extracted from the neural net, from the convolutional neural net, and the source response, which they define there as YST. So what they did is they combined the feature representation for all the layers, combined it, they stacked them all up for all the layers, they get this matrix. It's of size n by m. N is the number of images, m is the, depending on the layer, is different size. So they combine them all, and then they're trying to, for each layer, they're trying to predict their signal here, the source response. So you do all this matrix multiplication, and then you end up with a way to estimate beta. And they use this ridge regression, which I'm not too familiar with, but it's a way to, just a method to estimate these values. So for each of the source, they're the source dipoles, the source signals from the brain, this number has this many regression models. So, and with that, I'm going to hand it over to Arna, who will take us through the results and stuff.
Maybe just before I start with the results, is there anything that, like, any comments or any questions? Yeah, sure. Yeah.
Is there an advantage of using MEG over MRI?
Sure. So if you've read the paper, they talk about a paper using fMRI, which showed this. So fMRI doesn't have the temporal resolution, although it has the spatial resolution. So this work was using, MEG, but they also have a work using fMRI. So the problem with what they wanted to do in this paper is show that the representations change with time. So you can't do that with MRI, because I think the temporal resolution of MRI is around 2 seconds, is it right? Roughly? Yeah, so, and the visual processing happens around like 200 milliseconds, or, I mean, there have been papers that have shown object recognition, people can do it within around 100 milliseconds, 125 milliseconds as well. So you cannot capture that using fMRI. Raymond, you had a question?
No, I just want to say that
Regularize. Yeah. So it's to avoid overfitting. Okay, and so just to clarify on Andrew's question, so they don't have an encoding and a decoding model per se. What the basic contribution of this paper and why we wanted to discuss about this paper is because they've done a lot of work, and interestingly they want to compare, are the CNN representations for images same as the representations in our brain? And so the encoding model, what they try to do is they try to find a linear mapping or linear readout from the CNN representation to the MEG representation, or the representation in our brain. Why they are different, what is the unique point of this paper is, earlier what people used to do is they would show humans 500 images, they would show the CNN 500 images, form a representation similarity matrix that, okay, this image, how similarly is it represented like across sessions, let's say. And they would form like a 500 cross 500 matrix, and they would correlate the two matrix for the two species, let's say, I mean, if CNN is a species. So that is what gives them this. So this is the correlation scores for different layers of the CNN. So it's like layer 1 to layer 8. I think they use the same VGG net. And these are the areas which show like high correlation. So it's like the lower layers represent, let's say, the primary visual cortex stuff like V1, V2, stuff like that. And as you go higher, like more abstract layers where you have fully connected layers which lose the spatial information, you kind of represent more like abstract information, which probably is in the more in the ventral stream, so it's useful for object recognition. Yeah. So I think what they did was they also compared it to the null model, and these were like significant as far as I remember. But yeah, I agree, it's not a very high correlation. But in this paper, they also attributed to the noise in the signal and everything, because the mm-hmm. No, so in the null model, yeah, I mean, we're not going to the statistics for this, but as far as I remember, for the null model, what they wanted to do is, if you have a random permutation, or if, I think they do it with a randomly trained network. So that is to check if the CNN representations, I mean, if learning the CNN representations actually make it better. And the other thing that they did was, from the image set they had, at least in this paper that I'm talking about, not in the current paper, because in the current paper I don't remember the null model. It's probably somewhere in the appendix. But they kind of like changed the identity. So they shuffled the identity of the images. So image A shown in trial 1, they tried to see the representation similarity with image B shown in trial 2, rather than doing the same thing. So they kind of shuffled the representation matrix, and they tried to do the correlations. And so these are the ones that actually appeared to be significant. So yeah, but yeah, the correlations are pretty low. And there is another interesting paper which actually talks about this and shows that if you have that particular stimulus space, the representations are not similar. But okay, we can talk about that another time. So, okay, there are a lot of figures that we can talk about, but we'll talk only about a couple of them because those are the main contributions of this paper. So the mean encoding performance is, okay, how well can we actually decode, or how well can we encode the MEG responses from the CNN responses? So from their encoding model, the linear regression model. This is across different repetitions for each subjects. And this is mean encoding performance across all time points, across all sources. So the main takeaway is that, okay, for some subjects it does pretty well. For some subjects it's not able to do that well. And the main argument they make is, okay, some subjects moved in the scanner, so that could have changed the registration, and source reconstruction problems could have affected them. But yeah, the main takeaway, it works for some subjects, it doesn't work for some, so it's not very well explored. So in the figure, in the paper, figure 3 comes before figure 4, but I guess figure 4 should have come first. So figure 4 is what shows the main contribution of the paper, that which regions of the brain are well explained by which layers. So here again, correlation of 0.3 is something they got from the threshold with the null model, which they did. We'll skip that for now. And the color coding is done by which layer, like according to the layers. So the main the main purpose of using MEG was to see if there's a difference in the temporal information, which is something they show here. Let's say if you just see for subject 4. So the same region, the same anatomical region in the brain is explained by different layers if you go through time. So initially, after like 75 to 105 millisecond of stimulus presentation, it's probably the layer 1 and 2 that explains the V1 the best, or like the visual cortex the best. But the same regions, let's say this region becomes more greenish as you go further down in time, which is what they wanted to explore. And this, they talk about recurrent connections that feed back from higher layers to lower layers and effectively change the representation, and therefore change the neural activity in that region. So they say that even lower regions could be representing like higher level features just because of those recurrent connections. And this ties back very nicely to the literature of object tracking, where maybe if you're like in one-shot learning where you suddenly see a ball out of nowhere, it's difficult to see what, or if it's difficult to understand if it's a ball, flying bird or something. But once you see the ball, it's easier to track because you already have an information, a recurrent process going on that, okay, there's this ball, you have those features, and it's a higher level representation now. So this is again the comparison to the previous paper where they used representation similarity analysis. And this figure, again, this is figure 3. So this is how they judge like how well could we, this is the prediction and activity correlation. So the predictions they make from the linear regression model, they correlate with the actual recorded activity in the validation set. And so this again shows like which regions were significant, I guess. This actually shows the correlation. So here the correlations are stronger than in the representation similarity, as you can see, which I guess is expected, because when you're talking about a representation similarity matrix, you're taking into account that the same same stimulus can be represented in different ways because of the inherent noise in the neural responses and then in your recording as well. But here what they're trying to do is they're trying to predict on, let's say, a bunch of images that were shown, and then they try to train the model on a bunch of images that were shown in the training set, and then they try to predict on some images that they held out for validation. So the correlations are higher in this case, and it was more, they wanted to show that, okay, we can decode most of the visual cortex, or we can explain most of the responses in the visual cortex in this figure, they also talk about that how it changes. So this is the best activity that was explained. So if I go back to this figure, so let's say in this region, it's layer 7 that's best explaining this region. So here they would be showing the layer 7 activity prediction, layer 7 prediction using layer 7 features with the activity of this region. So it's the best one, so you can assume why the correlations are high. And I think this is an interesting figure that they show. So this is where they compare like which layers, when is the temporal onset of layers. So like layer 1, layer 2, they have a very quick onset. So because they're low level features like edge detectors, Gabor filters, as Raymond pointed out. So these layers appear very quick, appear very quickly in the explanation model. So I mean, layer 1 to 4 almost appear at the same time if you look at the median times. Layer 5 appears sometime later, and then layer 6 and 7 are which are the more abstract representations where you lose the spatial information, comes later. And they talk about that there's a significant temporal gap between layer 5 and the previous layers, as well as layer 5 and the next layer. So they call this like kind of a higher level representation, which represents like between the lower convolutional layers and the higher abstraction layers. They don't really talk much in terms of neuroscience for this, or they do any further kind of like analysis as to what is it that layer 5 is looking at, what are the features that layer 5 is finding out, but I guess this could be studied in further studies. This figure, it's kind of what we actually looked at, but it's kind of doing a parcellation. So talking about, so grouping basically V1 as saying it the primary visual cortex, V2, V3, V4 as early visual cortex. So more like grouping small regions of the brain into more coarser regions that is more colloquially known in neuroscience. And what they want to show again is that it's different layers that represent different anatomical regions at different time points, and it changes across time, which is their major conclusion from this paper, that because using fMRI or representation similarity, you cannot do this necessarily, because you're looking at, okay, during the time I was showing the stimulus, what was the representation similarity and stuff. This is an interesting figure because again, this is hinting at the same point, but they show that the regions that are explained by lower convolutional layers are explained by convolutional layers itself. So maybe this region, which is let's say explained by layer 2. So layer 6 doesn't necessarily explain this layer even in like later time points. And the regions that are well explained by layer 6 are not something that were explained by layer 2 in the earlier time points. So there is a discrimination between like higher level visual regions and lower level visual regions, even in time. So there are some regions that specifically represent abstract information for object recognition. There are some regions that represent very basic information. That's why layer 5 becomes important, because layer 5 kind of connects the two, and it can explain like both these regions. And then finally coming to the decoding part, which I guess, I mean, it would seem intuitive, but okay, the results are not as good. So briefly talking about how they do the decoding, it's kind of, let's say you have, it's kind of using the representation similarity matrix. So what they do is they have, this is the predicted activity for, let's say, image 1, and they try to correlate with the actual activity of image 1, and then they try to see, like, so this is the diagonal that you would want to see. So this is image 1 and this is image 1. So it's a weird way of representing rather than having it along this diagonal. So it's the anti-diagonal that this call as the major diagonal. So basically, since there's a major diagonal between a main diagonal that is highlighted with high activity correlation, this is you would expect that if you show like image 2 and the response that you get out of it, the predicted activity that you get out of it, probably the activity that you recorded is good enough if your model is good. So this is just showing that whatever we trained is actually useful, the linear regression model. We can actually use it for decoding. And they refer to some other papers, specifically an fMRI paper, which tried to decode movies from neural recordings. So the idea of that paper was they showed subjects some movies, and from the neural recordings, from the fMRI responses, they tried to decode what movie they were seeing. So I don't know if you guys have seen that movie, but it's a reconstructed movie from the neural responses. And I mean, a man riding a horse, the horse is running, or some video like that. It's pretty impressive, the reconstructions, but and they've extended that work using generative adversarial models and stuff like that. But the main idea is that, okay, whatever representations we're recording, they kind of have the information of what stimulus was shown there. So that's why they call it decoding. And yeah, that's the major figures of the paper. That's the major. So the major takeaway, again, for me, from an overall perspective, again, from the perspective of this reading group, is that they show that the representations are similar, for sure. Like some layers represent similar information as in the MEG recordings, but this changes over time. So if you allow somebody to see images for a long time, the representations keep changing. So that's the major takeaway from this paper, and they use it like they use MEG to compare the CNN responses with the neural responses. Comments or questions, or did I just zip through pretty fast? Yeah. Okay. Yep. So that's that. Yeah, sure.
So I guess one thing that I'm
Mm-hmm.
in general is
Mm-hmm.
it generates data coefficients
All right.
that the traditional
Don't work exactly.
is it just that the actual fact that actually delineates from, or like
So in this case, to calculate the p-values, they actually took, they did not use the beta coefficients. So they get this beta trained on the model, then they use it to predict on the held out set. So they get, let's say, some y hat, okay, which is estimated y, and they do a prediction between y hat and y. So Spearman's correlation coefficient. So once you have correlation coefficients, and you have a distribution of them, let's say you get a distribution even from a null model, then you can say, okay, what is from my null model, this is the distribution I get, this is the correlation value that I get, is it different or not? So that's the idea. Is that clear? Or because then you're not doing the statistics on beta anymore, you're doing it on like correlation coefficient. So then you don't have the problem of the scaling and skewing problem anymore, right?
is like a null model necessary though? If you have like a correlation coefficient that is high, is that sufficient to say that there is a
So it's always good to have a null model, because then you can say that, okay, whatever I'm seeing is not noise. It's not because of noise, because it could well happen that maybe you have a DC signal that guides all your responses, and then even if you shuffle around the sources time, you still have the DC signal across sources. So your null model it would still give you like high correlations, just because there's a DC offset. So it's good to have a null model. But I mean, there are many papers that show just the top 5% correlations or something, which says that, okay, these are the top 5% correlations that I have. And these were the best 5% sources that I could explain, or best 5% sources in time that I could explain. So yeah, there are ways to do that. But yeah, it's always good to have a null model, so your results are more convincing that it's not because of some inherent noise or some external variable that guides everything like at the same time, that's guiding the model predictions. Yeah.
Do you think it makes sense?
I mean, in this context, it does, but okay. So the idea is like correlation allows you to compare across time and across regions. So because even if you have like different SNR, so you can like do a p-value for that region if you use a null model for that region specifically across time. So let's say, so for example, like if you have YST, right? So if you want to, because each anatomical region would have its own SNR depending on how deep it is in the brain and stuff like that. So what you do is you only form a null model by shuffling T. So you have a null model for each anatomical region, and then you can do a statistics for that. So that's why it makes sense to do a correlation. But yeah, what like?
I don't know, I feel like you've got
Mm-hmm.
brain and deep
CNN,
learning.
yeah. Mm-hmm. Yeah, it's really not the whole story. I mean, what they wanted to see is, okay, I mean, from a representation from one particular layer, is there a linear transformation of those features to which I can predict MEG, let's say? So neural recordings. I mean, instead of doing true. Yeah, true. That's the fun part of it. Like they have first thing, I think they had like, I don't know, 12 subjects, I guess.
Yeah,
Yeah.
they're showing the
Top four.
subjects.
Yeah. So yeah.
And they're looking.
True. I mean, the results. Yeah, that's another thing. This paper, the results are not as great because first thing, they train the model on each subject. It's not like a model that is trained on cross-subject all data. So it's trained on, one model is trained on one subject, one ROI, one time point. So it's super possible that it's overfitting. I mean,
Yeah.
it's just possible.
Like the ridge regression.
Yeah, that's
Value of that regularization.
a lot. Yeah, exactly. So. So yeah, that's why.
Dynamics are hard.
Yeah. And actually, like there are a couple of papers like this that show for different, like different stimulus set, different modalities. They show the same thing, that okay, it's possible, it's possible. But then there's one paper from last year, I guess. Yeah, 2018 December, I guess, from the MIT, James DiCarlo's lab. So they showed it's Rishi Rajlingam. He's the first author. So they showed that if you actually constrain your stimulus space to be as same as the stimulus space on which CNNs are trained, then the representations are not same. So what they do is, that's bad. Yeah, that's super bad. Yeah. So it's but again, I mean, I haven't read the paper thoroughly, but that's their main like message, that it's not the same for all stimulus space. What stimulus space you're seeing, probably that has a big role in guiding, and it makes sense because what you're doing is all of this analysis specific to the stimulus, to the 50 images that you showed. Representation similarity is specific to those 50 images. The correlations are specific to those 50 images that you showed. It's hard to make a generalizable statement about that. Okay, any image you see, CNNs do the same thing and humans do the same thing, which is a big claim in this paper, which they don't really. Yeah. And not just this paper, this family of papers. So. But yeah, that's something that hasn't been addressed in the field so far. So it's a big open question that, I guess, because these papers came out earlier in time, so in the deep learning community, it's more or less accepted, okay, whatever we are getting is similar to neural representations. But the story is not that simple. Maybe most, some, yeah, those who do care, they think that, okay, it's similar to neural representation. So we're actually having a model like Kendrick K and their group. So they have this paper, opinion paper on using DNNs or deep neural networks as model organisms. So they call it D-animals or something, yeah, D-N-I-M-A-L-S. So yeah. So they talk about, okay, if we have this model similar to the human visual cortex, we can do ablation studies, we can do stuff like that to see what happens, and talk about visual deficiencies. So it's a big field that way, but the problem is the claims that they're resting it on is not that well established. Okay. All right. So thank you everybody for coming again. And all right. Sure. So I think the next meeting we'll be having a presentation, somebody presenting their work, because that's how we plan to do it. Presentation in one and a paper discussion in the other meeting. So we are planning to do that alternate. For the paper discussion, we would really like if you guys have some suggestions for papers that we can discuss. This was a paper I just picked up because I felt like there are a lot of things to discuss in this paper. There are a lot of good things, there are a lot of bad things that we almost covered most of it. So it's good. It would really help if you can send us emails or reach out to us on the blog, make issues on GitHub, whatever. Like just get to us with any suggestions of papers that we could discuss. And what we would try to do is before the next meeting or just after the next meeting, we can have a voting period where you guys can vote that which paper, if we have like a couple of choices, which paper we will discuss. It's not that if your paper gets lost in this round of voting, it wouldn't win. Anything can happen. So we'd discuss it in the later meetings for sure. Elections, so anyways, yeah. So because we feel that it would be more, we can have more discussions if you guys contribute as well, rather than we telling you that, okay, this is the paper, because sometimes, like when we choose a paper, we have read it, we get the takeaway message, and we say that, okay, this is a takeaway message that is worth discussing, but like for this, I knew the takeaway message, but when I was going through the paper yesterday night, I realized that, oh yeah, there's a lot of things that we don't want to discuss, which is probably not that evident from the paper itself. So yeah, we can all we can work as a group. We can discuss we can vote for which paper to discuss, and accordingly have a better discussion next time. All right, thank you.